Well, thank you very much, and for that very kind invitation and kind introduction and invitation. It's really a pleasure to be here. I'm going to start by just sharing my screen and make sure everybody can see the slides. So can folks see slides and hear me? Great.
Yeah.
Good. Now I've got to get my my prop. I just want to say it's lunchtime over here. I've saved my apple. And I suspect it's getting towards pub time or happy hour or however you call it over there. So thanks to everybody for doing this and being with the talk. Great pleasure to be with you, and you'll see that some of these topics that I'm going to talk about are things that many of you are familiar with, and in many cases will know more about than I do. But today, let me just move forward. I want to give you my disclosures. First of all, I'm involved in a number of areas, including my wife is part of a startup that I'm not going to talk about today. Those areas that are in yellow are things where I have some conflict of interest that I'll be talking about, and I'll try to disclose that as I go through. So today I'm gonna give you three themes about whole body PET, and I'm going to start by why whole body PET. And in particular, rely a little bit on the background that you heard about with me as being an instrumentation physicist for a while. And then I'm going to move to my clinical background, and in a session I call Fun with Total Body PET, and show you some of the things that we've been working on. And then finally, I want to move back to the science a little bit and go to cancer metabolism, and in specific breast cancer metabolism, and show you some of the work we've been doing in that area, including a multi-tracer injection with whole body project that was sponsored by the Biden Cancer Moonshot, which you may have heard of as an active program in the U.S. And so I'll take you through all of these processes in about 40-45 minutes, and look forward to some questions afterwards. So let's start with theme one. So I suspect this slide is not needed with the crowd I have here, but just to remind you, what we're talking about here is PET. We start with a cyclotron. That's our IBA cyclone that's in the basement of our nursing building. We go to our gleaming GMP center. We make a radiopharmaceutical, FDG for our clinical practice. We inject that into a smiling patient. You can see my patient drawing. I better keep my day job and not be a medical illustrator. Then we inject, go into a PET scanner these days, typically a PET CT, maybe a PET MR scanner. And when we're all done, what we get is an image of tracer concentration. And from that image of tracer concentration, we can do some quantitative analysis. And the fact that this is an image of tracer concentration, I think, is important, because not only does it give us pictures that we can interpret, but it gives us numbers that are important and can reveal everything from a more detailed understanding of what we're looking at to really insights into in vivo biology. So why total body PET, other than to keep people like me who are instrumentation physicists busy? Well, there's two reasons. One is you get better images. These are big devices and do a good job of capturing more and more of the photons that come out of the body from total body PET. So you can get improved visualization of cancer, and I'll warn you, I'm going to talk almost entirely about cancer today, and this will be a somewhat overly University of Pennsylvania version of the total body PET scanner. I'll be acknowledging those biases. But you get improved visualization, you get faster scans. You can do that half of the body we did in about 2 minutes with very high quality imaging. And you get lower dose, which is going to be important for a variety of reasons, and maybe especially important for pediatric imaging. But equally importantly, you get better quantification, because as you're collecting better quality data in these devices, in part, good part, because you get more counts, this not only gives you better pictures, but better quantification. Now, besides just giving you a prettier picture, one of the things that you can do with the total body PET that you just can't do with other scanners is look at simultaneous whole body imaging. You can use that to look at whole body kinetics and system biology. You can use this for dosimetry. I'm showing you a diagnostic example, but you could think about it in the theranostic sense and really get very accurate estimates of regional dosimetry, including in the tumor. And then as I'll show you at the very end of our study, we can go back to something that's a little easier to do with SPECT, and that is to have multi-tracer studies to give you multivalent, if you want to think about it that way, PET studies. So let's talk a little bit about the history of the evolution here. And here I want to recognize my mentor and longtime friend who died in the last year or two, Gerd Muellner, was a real pioneer in this area. And when I was first getting started, Gerd had recognized the need to move from these single slice PET scanners that were built in the 70s by some really pioneering folks. To these stack slices, which we were all using for a while, that use septa to prevent scatter, but limited the amount of activity that you could collect, to true volumetric imaging that allowed you to capture everything out of the patient that hit the detector. Now, at the time, that volume wasn't that large. These detectors weren't that wide, but this was an advance that Gerd and others at Penn and others working in the field made this advance in the late 80s, and this forms a fundamental necessity for total body PET. I do like to show this picture back when I looked a little different. I was fortunate enough to be a graduate student, as was mentioned, with Gerd and Joel Karp and Margaret Dobby Witherspoon. Joel and Margaret remain active in our program, and I'm here being a clinician instead of a physicist this time, but think back to my physics roots. So one of the interesting things about this early development, which we called the PennPET, and was actually first built with sodium iodide detectors, was that it provided, by modern standards, probably not wonderful image quality, but at that time, the ability to do true 3D volume imaging and really nice pictures of the whole body or whole body torso. My job as a graduate student was to take these sodium iodide detectors and get them up to count rate for PET scanners. And later, this approach was replaced by lutetium-based detectors, LYSO, that had some of the same good light output properties that gave you good energy discrimination to reject scatter, but also had much faster timing. So you can see a picture of what that looked like. And again, I want to recognize not only Gerd, but Joel Karp and Margaret Dobby Witherspoon for this work. Now, another major step in the path towards modern and total body PET scanner is time of flight. And I'm picturing Suleiman Sirti, who was one of the people that led the resurgence of time of flight. I'll go back, show you some history on this in a second. And this is probably something that people who work in PET now take for granted, but it's actually a relatively recent development on a practical scale. And just to remind you about this, when we don't have good timing information, the best we can do is identify a line defined by the two detection points of the annihilation photon components along which that photon must have occurred. And we go back to back projection or other related algorithms that fundamentally just back project or smear that activity along the full line. However, when we have time of flight information, it's not going to tell us exactly where that positron emission was, but it's going to get us close. And it's going to get us closer when that timing resolution is better. And as you can see, as illustrated in D, that can help separate out information. And rather than starting with a smeary blob that you've got to sharpen up, you actually place the information in much closer to its correct site of origin. And that dramatically improves image quality. Now, what has been shown, and I refer you back to this very nice review that Suleiman put in the Journal of Nuclear Medicine a few years ago, is this looks like increased sensitivity of the system, increased ability to detect the counts. And if you take the timing resolution down into the modern era, into the 200 to 300 picoseconds, you can see that your equivalent counts goes up by a factor of sometimes 10 or 20. That's like having 10 or 25 more times activity in certain ways. And so that's one of the reasons this is very important. The other thing the time of flight does you is these are very fast detectors. So for those familiar with PET principles and know what happens, that when you run at very high count rates, you get a background of random coincidence. Because these detectors are so fast and their timing is so good, they don't have as much dead time, and they actually are less susceptible to applications. It's important to remember that this was an important development in PET. Just wanted to do a check. I have a couple of things flashing on my screen that says I lost connectivity a little bit. Can everybody still hear and see me?
Yeah, I can hear you.
Okay, good. All right. There's probably a little bit of a delay between my pressing and what gets to you. Now, this is not a new idea. I'm showing you Michelle Terpagosian, one of the real pioneers in PET overall, who is at Wash U. As a matter of fact, we had a Wash U speaker last night who was a Terpagosian professor. And what Michelle did was to really recognize the ability of time of flight and leverage what was state of the art at the time, which was barium fluoride or cesium fluoride detectors in analog electronics with individual photomultiplier coupling. And he was able to show that with time of flight, you reduce the noise component of the images quite a bit. The problem is with analog electronics and these one-to-one couplings with photomultiplier tubes, this system was hard to keep stable. We actually had one of these super PETs developed by the Scanatronics company when I first got to Seattle. And it was really a beautiful system when it worked and when it didn't drift, but that was hard. Now, more recently, in the 2000s, Suleiman and Joel Joel Karp and Gerd Muellner reintroduced this at a time that there were still photomultiplier tubes, but fast photomultiplier tubes. This has now actually been replaced further by digital electronics. And again, with the Philips system that was the first one to release this sort of revamped time of flight commercially, you can see how much less noisy these images are with time of flight. And what this turns into is much improved both qualitative and quantitative image characteristics. And this is just from some of the work that came from that, where there was improved lesion detectability with time of flight versus non-time of flight. This was especially important for those patients that were large enough to attenuate a lot of the photons. And one of the things that time of flight does is makes your image quality both quantitative and qualitatively consistent across different patient sizes. Everybody can take a good picture of a skinny patient, but the larger patients really benefit from the time of flight, as shown in these graphs. One of the other things that is not completely understood is that this gets you a lot closer to the answer to start with. And so as we're all used to these iterative reconstruction algorithms, and I'm showing you on the x-axis the variability of uptake and the ability to recover the uptake, the time of flight moves you into needing much fewer iterations, taking you to much lower noise levels to recover the spatial resolution, and is much more efficient. So the time of flight is better quantitative accuracy, both in terms of reduced bias and improved precision and decreased noise. So that's where the field stood from the early 2000s up until probably about 5 or 6 years ago, where this consortium of great minds got together for the Explorer project. And a lot of the inspiration behind this are names that will be familiar to you: Simon Cherry and Ramsey Badwai at Davis, Terry Jones, who's not too far from you in Manchester these days, and then Joel Karp, who you've seen before. And these four formed the cadre along with others. I was pleased to have some involvement in this Explorer consortium. And Simon and Ramsey were able to get a relatively large grant from the US National Institutes of Health to be able to put one of these systems together. And to the great credit of Simon and Ramsey and the full group, when they started in Davis working with a Chinese company, United Imaging, they found that their development costs were a little bit lower than they had anticipated, and offered the ability to make 2 of these devices and build the second one in Joel Karp's lab, which is probably one of the few academic labs that can take the process from start to finish. And so not only were we able to build these 2 devices, we were able to build 2 rather different devices, one with a slightly different technology, and number 2, one that was a commercial device, and the other one was much more of an academic and commercial partnership. Now, first out of the gate, before I get there, let me just explain to you why this is important. When we have a conventional PET-CT scanner, even when we have a large field of view, we have to get to the whole body by stepping through that patient or moving the patient back and forth. The idea of the whole body is you capture the whole body at once. You have an increased sensitivity, and you don't have to move the patient to capture the dynamic bio distribution. This particular illustration is actually showing you what's now commercially available. Siemens is making a camera called the Quadra, which has a 100-centimeter axial field of view. But this idea was revolutionary back in the mid 2010s, around 2015, when this concept was being developed. Now, first out of the gate was the Davis group, and these are some of the extraordinary pictures they have shown in their 2-meter long system. They truly are right now the only fully whole body device. And I refer you to a number of their publications, which will show you the extraordinary capabilities of these images. And these were really the first human images that were a little bit, a couple months ahead of where the Penn group was. I'm now going to shift over to the Penn effort. And one of the things that was distinct about our effort is this was built in pieces at a time. And that was on purpose to make this a scalable device that could go all the way up to our maximum width, which right now is where we are at 140 centimeters. That gets the head all the way down to the knees, and sometimes the whole body in shorter people, but allows a scalable system that could meet clinical needs and might be, for example, a shorter system for a pediatric population or a more clinical operation. This has 4mm spatial resolution, so pretty close to the state of the art for the detectors, 235 picosecond time of flight resolution. If you're not used to those units, that's pretty good. It is close to the state of the art, not quite at the state of the art, and importantly has good energy resolution so that this wide open scanner can reject scatter. There's a lot of technology development in this. Part of this was a collaboration with Philips, who provided some in-kind donations of detector material. This is now the modern era where we are using not photomultiplier tubes anymore, but silicon photomultipliers, which are solid-state devices. You can see how compact these are, what the detector module looks like, and the relatively large effort in electronics and data processing to take the gadzooks, the ton of data that you're taking out of this, acquire it and reconstruct it. And so this is what the scanner looked like in various stages of its construction. And as I mentioned, there's a time of timing of 240 picoseconds. And this is what it looks like in the more commercialized process, where we now have the full axial field of view scanner coupled to a CT at 140 centimeters. And this is, if you took the covers off, what it looks like with 3 rings or 6 rings. And here's Joel and the many people in his group, some of which had industry experience before, together with Gerd and others, really helping to put together this really exciting device. All right, so I showed you how we got there and dragged you through a little bit of physics. Let me now show you some fun with the total body PET scanner. And again, I'm going to focus primarily on what's been done at Penn and some of our recent studies. And so this work that I'm going to show you includes not only a consortium with Joel's group and his postdoc, Varsha Visnawath, you see pictured on the right, but 2 of our really gifted clinician investigators, Dan Prima, who is the head of our PET Center, and our nuclear medicine division chief, and one of our younger faculty, Austin Pantel, who comes with a math background and has really enjoyed being at the interface of math and clinical work in this case. So here's some examples. We have a Philips Ingenuity scanner, not quite state-of-the-art scanner, but pretty close to it. Gives us good image quality. That's our research device. And so you can see what happens that when we move from a 20-minute vertex to thighs image to almost that same field of view in our 70-centimeter prototype, we get really dramatically increased image quality. And in fact, we get the same image quality, maybe a little bit better, by taking a 2-minute scan. So these are extraordinarily sensitive devices, to the point where with a conventional step and shoot type of approach, you wouldn't get the same image quality. This also leads to increased detectability in lung cancer, again, work that Dan and Austin are working on, where you can see how much more prominent this small lesion is in the lung on the Explorer compared to the background of noise, and actually reduced contrast of the standard of care PET, which is a pretty decent time of flight standard of care PET. This also works not only for F18, but for other tracers. I suspect you also do a lot of gallium-68 DOTATATE for your neuroendocrine tumors, or another SSR tracer. And you can see that 3.5 hours post-injection, which is a quarter of the activity that we image from 1 hour post-injection, you can see the extraordinary image quality and clarity that you get in this situation for short-lived radioisotopes. This also works equally well for long-lived radioisotopes. Mike Farwell at our place has been working with immune imaging, including the compound from Imaginab, their CD8 imaging agent. It's zirconium, so when you give zirconium, as you know, you can't give that much of it because it's a pretty high radiation dose. I think the injected dose is typically a millicurie or less. For this approach, and again, you can begin to see that that you have much less noise and much higher clarity in this situation, where you're again working at lower activities for different reasons than you would with a conventional scanner. Now, what about that whole body capability that I told you about? Mark Selmayr is working on infectious imaging agents and is working with a trimethoprim labeled trimethoprim, C11 trimethoprim, and more recently an F18 analog. And so this is, when you're doing your dosimetry in the study, this is not somebody that has an infection as part of our normal dosimetry. We can go out to 6 half-lives of this tracer and connect a fully dynamic biodistribution that will not only inform our approach to kinetic analysis and image analysis, but provide us with really a wealth of information about dosimetry. Now, when we're doing these diagnostic agents, we like to get accurate dosimetry, but I show this to illustrate that when you're in the theranostic range, where you're doing paired diagnostics and therapeutics, the ability to take this kind of image will really give, I think, extraordinary information about regional tracer uptake and therapy dose that will allow us to calculate not only normal organ doses, but calculate tumor doses, which is what we should have been doing all along. Paco Bravo, who runs our nuclear cardiology program in our place, has been working in a large animal model. He's been working in a pig model and looking at conventional perfusion imaging with rubidium, as well as some novel imaging agents. And you can see the extraordinary detail you have in the flow phase portion of his studies, where you can really get a nice view of the vascular tree, and you can see the type of image quality that you're able to get over the standpoint, including with both rubidium and F18. So there's some other insights that we can get, and I want to come back to our friend FDG, and just to remind you that this is a tracer of glucose metabolism. Like glucose, FDG is transported into cells with similar transporters like glucose. It goes through one of the rate-limiting steps of hexokinase and is phosphorylated, but unlike glucose, it can't go any further. Now, and I'm showing 3 of the folks that have played a large role in that. Lou Sokoloff is a pioneer at NIH that developed the deoxyglucose technique. His then fellow Marty Rivich moved to Penn, and together with the Vassalabi implemented some of the early work, I think, including maybe the first FDG scan that was ever done. One of the controversies that comes up in this place, in this setting, is the reverse direction, because we know that some tissues, such as the liver and the brain and muscles, can actually dephosphorylate glucose because they have to make glucose either from gluconeogenesis or the breakdown of glycogen. And there's always been a fair amount of controversy into what extent that happens with FDG, where there were some images that suggested there was some efflux, but it's sometimes hard to tell. Well, when you have this kind of device, you get some really interesting insights. I think this may have been the second patient we haven't ever imaged on the scanner. And we see that patient at 10 minutes, a little after the typical time of 1.575 hours, at 4.3 hours, and then at almost 19 hours. So you're almost 10 half-lives down on this tracer. So what do you see? You see that unless you're listening to my talk, in which you may be falling asleep, your brain's using most of your glucose. Fortunately, this subject cheated a little bit on his diet and had uptake in FDG, which we only see if you're in a slightly fed state. And we can see that over time. But if you look at those 18-hour images, you can see that the brain has largely washed out and the heart hasn't. So the brain, as expected, can dephosphorylate FDG because it can use and break down glycogen. The heart cannot. At least not to the same extent. And yes, we kind of knew this from kinetic analysis, but there's nothing like seeing this. And I think this is going to provide us with some really unique insights into biology. Now here's our version of that fancy picture. If I had a little bit more courage, I would show you this as a movie, although sometimes it doesn't work. And you can see what looks almost like an angiogram in the early dynamic studies here, giving you the extraordinary kind of quality that you can get from these types of images. But unlike the typical angiogram, it's not only pretty pictures, it's quantification, and you can see a faithful reproduction of different vascular pools. You can see that double hits we sometimes see in the injection, and then the dispersion of the bolus as you go further and further away from the heart and out to the peripheral circulation. And you get beautiful kinetics across all the different organs. If you wanted to do whole body and system kinetics, you have everything you need to do that. So we think about some of the translational applications, and this comes from the Davis group, and showed you this picture before. You have all of these time activity curves. You can couple this to a compartmental model, which I also like to do as well, and really change the approach to the way we analyze PET. Not only take pretty pictures and look at input function driven measures of kinetics, but really look at whole body kinetics almost the way a bona fide pharmacologist would look at it, with tools that most bona fide pharmacologists have not had. Now, in this situation, we need quantitative information over time in these dynamic images. And this is where Varsha, who has put these in a couple of locations at this point, ran some simulations using a technique that had been developed by Suleiman Serti a while back. And that is to take real patient data, which comes in a list mode stream, embed these fake objects that we have imaged in our system and develop them, insert them into the list mode stream, generating something that looks like a real image with known lesion locations, as you can see here at various times. So what Varsha did was to go back and look at a full dynamic semi-simulation here. And what you can show is that in the whole body, over a variety of regions with different levels of uptake, we actually get quite a bit of precision in this area, not only in high count regions such as the liver, but in some of the lower count target to background regions in the lung. So again, it's not only pretty pictures, it's accurate quantitative data, which is an important part of this process. Now, there's some new tools for this. Samuel Matej is a reconstruction expert at our place, has gone back to an idea that's been around for a while, but is now done much more powerfully with an algorithm that he's developed called DIRECT, which is a very efficient iterative reconstruction algorithm. And what you can now do is embed the time characteristics of the tracer in a inside the reconstruction algorithm. And so this is a simulation where this was made with a linear increase over time with a time activity curve that you can see. And that time activity curve was part of the fitting process, part of the constraint process, in addition to the physics of the problem. And again, you can see the dramatic improvement in image quality, especially noise in this situation. As part of the work that we did for our cancer moonshot and working with Despina Cantos, who is an expert in AI and radiomics, also works in breast cancer at our place, and her brilliant graduate student in bioengineering, Rhea Tatalia, took this all the way to true 4D AI and radiomics. And without taking you through all of this, went in and used some phantom simulations to identify approaches to identifying principal components in an approach that she calls functional tumor heterogeneity, and then taking this and deriving parameters based upon radiomics and that feature. So it's actually blowing up our compartmental models and letting the computer figure out what it's looking at. And in fact, in the first try, she took a data set that I had been working on for only about 20 years, and through even sophisticated kinetic analysis, had never got it fully predictive, whereas at the first try, this model was able to identify an improved prediction of downstream outcome from these FDG PET-CT scans that had not been possible with conventional imaging. This was published recently in the European Journal of Molecular Nuclear Medicine and Molecular Imaging, and there are a number of other folks working on this, and I think it's very exciting to think about the combination of our 4D kinetic approaches and more modern data science driven approaches that will change the field. Now we can also use these simulations to be able to use the whole body device to get a new approach to trying to get more information out of our scans. Lisa Dunwald, who's now at the University of Iowa, was with me at Penn for a while and with me in Seattle for quite a bit, has worked on dynamic imaging in our breast cancer studies. And to make a long story short, we showed that full kinetic imaging provided estimates of flux and transport of FDG that drastically improved our ability to predict outcomes in terms like survival and relapse in breast cancer that was treated with pre-surgical chemotherapy. However, this required the patient being in the scanner for a full hour at one region in field of view and then moving to a whole body static scan, which I have misspelled slightly there, to generate the time activity curves. Can we do this? Can we do this differently with whole body scanners? So we're back to Varsha's work, who took the current protocol of a 60-minute dynamic scan and then tried a couple of different scenarios where she thought maybe the full dynamic scan would predict the later scan, or maybe we can do a flow phase akin to what we do when we do a 3-phase bone scan and take a short dynamic scan followed by a PET static scan at standard times. And so in return for what might be a relatively short early flow phase study, get this full dynamic imaging. And so again, she used simulations and found that strategy number 1 didn't work all that well, as it was hard to use the dynamic PET scan to predict what was going to happen at the end of the scan until you got close to the scan. However, if you got the early portion of the scan and then skipped a bunch and then took a static scan at the end, that actually worked quite well. And as you can see, depending upon how much precision you want from your flow phase, you might be able to get away with a flow phase as little as 10 minutes, followed by a pretty quick static scan at the end that all of a sudden makes dynamic PET imaging feasible, where you can measure not only an SUV, but measure delivery and retention of the tracer. And you can see what that algorithm might look like. Okay. I'm going to give you a chance to take one more breath before I hit my final stage here. And I want to come back to a theme about cancer metabolism and multi-tracer agents, and my new math, which says 1 and 1 is greater than 2. And here I want to go back to some of the other mentors I've had in my life, Ken Crone and Janet Erie. We're all worked together for many years at the University of Washington in Seattle. Ken is now a little south in the University of in Portland, Oregon, at Oregon Health Sciences University. And Janet actually now runs the NIH Cancer Imaging program as an associate director. So Ken had posed a couple of articles that I had the pleasure of being part of about multi-tracer studies. This started by what he called true tracers, comparing analogs that are highly metabolized, carbon-11 glucose, carbon-11 thymidine, and comparing them to their less metabolized analog, fluorodeoxyglucose, and for those familiar with proliferation tracer, fluorothymidine. And what we were able to get from here is a number of insights. Number 1 is when an FDG was first developed and deoxyglucose was first developed, there was this a concept called the lump constant, which related deoxyglucose or fluorodeoxyglucose measures of glucose consumption to those of the native substance. In pioneering work that was done by the late Alex Spence in brain tumors, he showed that that constant works for the normal brain, but not for tumors. And there's no such thing as a normal tumor that uses these tracers in proportion. We're able to take the same approach in a paper that Janet published, working together with Finbar Sullivan, who's now at University College Cork, and we're able to gain insights from looking at thymidine flux using native carbon-11 thymidine and applying that to FLT to be able to do a much better job of using the tracer in the brain, where the blood-brain barrier makes life complicated to predict outcomes in glioma. Now, a little bit later, we revisited this with multiple imaging probes to the point where you begin to see how different tracers might be helpful. One of the areas I've spent a lot of time in, dating back to our Seattle days, is in looking in breast cancer estrogen receptor expression with FES, which has been recently approved in the US, and FDG. This is a very useful combination because the FDG tells you where active tumor is, and the FES tells you whether or not it expresses estrogen receptor. It's actually a very valuable technique. It's just klutzy when you got to do that on 2 different days, and this would allow you to do it on 1 day. Janet has done some fascinating work looking at multiple tracers, metabolism, blood flow, proliferation, drug transport, hypoxia, to gain some insights into sarcomas, which are some of the most complicated tumors, as those that you work in this area may know about. This had me go back to work that I had been doing for quite a while, and I want to credit my former University of Washington group working in breast cancer. There's a bit of a diaspora now, a couple of us here at Penn, where we went back and looked in more detail at the kinetics of FDG and perfusion measuring using O15 water in breast cancer that were larger and locally advanced that were treated over the course of therapy. And what we found was that perhaps not surprisingly, glucose metabolism, or at least hexokinase flux as measured by FDG, by the FDG-Ki, really didn't bear any relationship to blood flow in breast cancer. And there wasn't a reason to think this, that metabolism is disordered, the blood vessels are not particularly regulated. Whereas if I had shown you the same fit with real raw data in the normal breast, you would have seen a reasonably tight line in that case. Now, does this make a difference? Well, it kind of does, because I'm showing you two examples where you have a tumor that is really running hot and using glucose like crazy with relatively modest blood flows. So it's using pretty much everything that's delivered to it, versus that image at the bottom, which quantitatively is pretty well matched and falls a little bit closer to the normal breast line with the expected level of glucose consumption matched to its level of blood flow. And I use the analogy. So number one is, why is this important? Because that mismatch, that tumor that was burning hot, even though it looked like it had a good response, the patient didn't do well and had early relapse and death. That other tumor had a pattern resulting in a complete metabolic response, and the patient went on to be disease-free, and to my knowledge is still disease-free. So there's something different between that fuel guzzler that's using glucose like crazy. For those of you that are not familiar, this is a Hummer, a car that uses a lot of gas and are fuel efficient. I think this is a Honda Fit that uses its fuel a little bit more moderately. And I'll come back to that theme again in a second. So fast forward about 10 years, and GenSpect reanalyzed this data using a more modern approach to breast cancer subtype. And as many of you may know, the extremes in this are those ER-positive tumors that look like the normal breast epithelium, as well as those triple-negative tumors that come from the myoepithelial layer and are triple-negative in that they don't express estrogen receptor, progesterone receptor, or HER2. Now, in those ER-positive tumors, they looked a lot like the normal breast. They actually had relatively matched blood flow and glucose metabolism. And in fact, those more proliferative tumors that were using more glucose and had more blood flow were actually in the solid line of likely to have a good response, whereas typically the more indolent estrogen receptor positive tumor doesn't respond well to chemotherapy. The triple negatives reproduce that scattergram that I showed you before, and unlike the ER positives, having very high metabolism, especially in the absence of in low perfusion states, was not good. That mismatch of a lot of glucose consumption per unit blood flow was a predictor of bad outcome no matter what happened in the chemotherapy. And so we come back to our fuel efficient versus gas guzzling phenotypes. And in fact, this is something that I've been chasing since 2008, 2010, 2005, when we first started all of this. So in moving to Penn, I was lucky to come to a place where there had been a lot of work looking at alternative fuels with successive cancer center directors, Craig Thompson, who's now at Sloan Kettering, Chi Dang, who's now across the street at Winstar and runs the Ludwig Cancer Research Institute as well, in looking at glutamine as an alternative fuel. So for those that don't remember the cascade, this is a very highly simplified approach of glucose metabolism through hexokinase, branching at pyruvate, going into the TCA cycle. And then through the TCA cycle, it not only provides energy by going around the TCA cycle, but it provides molecules for biogenesis. A discovery by some of the folks I mentioned was the idea that many of the most aggressive tumors that also use glucose will also use glutamine, the most abundant circulating amino acid, as an alternative fuel, with the idea that that's a direct pathway to provide energy for molecules and biogenesis. So in the analogy that I showed you before, the low octane gas is the glucose, and the high octane stuff that gets right into the heart of the fuel system and provides energy in these molecules is glutamine, in my overly metaphorized, or whatever the right word is, analogy. We have FDG PET to look at glucose metabolism, and we have some glutamine PET compounds that look at glutamine metabolism. And here, one of the rate limiting steps, and probably the essential rate limiting step, is glutaminase, which turns glutamine into glutamate. And so we're going to focus on that as we go. Here's where I was very fortunate to be at a place with wonderful chemistry. Hank Kung, who's now emeritus here, has developed many, many tracers, including both carbon-11 and F18 analogs of glutamine. And when Bob Mack moved here shortly after I did, to head our radiochemistry program, he brought along some great talent like Sharon Lee and Prashant Padakanti that were helping to make and support this research. I'm going to focus mostly today on the fluoroglutamine analog. This is an interesting compound that's a little different than what we look at for FDG. It is phosphorylated in proportion, not phosphorylated, it's catabolized in proportion, it's transported, start over again. It's transported in parallel, both in the reverse and forward direction to glutamine, but it's not metabolized. And so it's very different than FDG, is that you're not seeing the metabolized down product of what we saw before. And this had some really exciting early data. This is work from Memorial Sloan Kettering, both preclinical model and clinical patients with brain tumors, showing the ability to visualize tumors with this a non-metabolized glutamine analog that were not well seen by FDG. About the same time, I started working on this with Rong Zhu, who's been a great partner in this process. And we looked at this from a slightly different angle. There has been a lot of work in trying to develop selective glutaminase inhibitors that would block the ability of tumors to use glutamine. So if they were addicted to glutamine, they would starve. Here's a number of the compounds. The one that has made us furthest to the clinic is CB839, made by the Calithera Corporation, that's been in a number of clinical trials. And by blocking glutaminase, the idea was to choke off these tumors that were using a glutamine and make them more susceptible to death or die on their own. Now, how could we interact with this? Well, what had been shown by the company developing and then repeated again in our group is if we block glutaminase, we are actually increasing the substrate concentration. Those tumors that are burning like crazy this widely abundant amino acid are taking whatever comes into the tumor and burning it and putting it into glutamate and possibly into the TCA cycle. But if we shut that down, their glutamine concentration actually goes up and their glutamine concentration goes down. And so a lot of our work is focused on developing pharmacodynamics of glutaminase, which is a real clinical need for translating these into the clinic. So we had done this, and as a matter of fact, this is just another slide showing that if we block glutamine metabolizing triple negative tumors, we see an increase in the glutamine concentration post-therapy. And when we give this non-metabolized tracer, we have a result that a lot of people are not prepared to see. They expect to see the glutamine go down. But in fact, because the glutamine pool size goes up and it's not limited by transport, we actually see an increase in the glutamine concentration pre and post inhibition of glutaminase, and in fact an increased ratio that is now characteristic and actually conforms to those types of kinetics. So this is an insight that came from our early preclinical work, and I think a lot of people are still getting used to this idea, and we have more work to validate it. But it shows you the way these non-metabolized tracers can work. And again, because I can't resist the kinetics, I just want to show you that when you give an analog that goes through this through the rate limiting enzyme such as glucose, and you're looking at the product, you'll see that product increase over time with higher activity. When you're looking at a non-metabolized analog and you're actually looking at the pool size of the substrate, it's a very different answer. And when the enzyme is active, the uptake is low, and when the enzyme is inactive, the uptake is high. This has now been validated with kinetics in mice, and I'll show you in a second in patients. So this worked for looking at differences in phenotype. Again, I'm showing you another triple negative tumor that had the blockade of glutaminase result in higher uptake. And in ER positive MCF7 xenograft, which does not have much glutaminase, uptake was higher to begin with and stayed high afterwards. Now, if this rings any bells, we actually use some of these very similar amino acid analogs, including fruciclovine, to look at these low-grade tumors that don't metabolize much glucose and probably don't metabolize much glutamine, such as early-stage prostate cancer that's non-castrate resistant or lobular breast cancer, right? Gas guzzler has a pattern of low glutamine that increases after glutaminase inhibition, and the fuel efficient non-glutamine user doesn't do that. And just to show you, this is now translated into the clinic. Here is a patient who had an ER positive tumor, and despite having a relatively small tumor that didn't show up that well in FDG, you can see it quite prominently on the scan, despite a small size, and it fits with some variability the type of model that we had seen in the mouse. All right, so we're coming close to the end of the discussion, and that is, how do we translate this into the clinic? How do we visualize this with a set of tracers? Well, we really need two tracers. We need to identify those tumors that are burning both glutamine and glucose. And with the tracers that we're using, we would expect to see high FDG uptake and not much glutamine uptake to begin with. And then with glutamine inhibition, we might expect to see small changes in FDG uptake, but we would actually see a profound change in fluoroglutamine. And so we're looking for a phenotype that starts as low FDG, high fluoroglutamine, and moves with inhibition to high glutamine metabolism and low FDG. Now, you go back to and say, okay, who cares? Why do you want to do this other than finding more things to do with total body PET? This is from a recent Nature article from Vanderbilt that looked at the uptake of FDG and fluoroglutamine in cellular constructs, and in fact showed that when there were certain conditions met, these had a profound impact upon the ability of the immune system to attack the tumor. And in those tumors that they showed in this particular cancer model that had relatively low glutamine uptake and moderate FDG uptake, versus neutrophils, which had a lot of FDG uptake and also especially low glutamine uptake. This impacted the immune microenvironment quite a bit, and in fact, inhibiting glutamine in this setting in studies that have come out of Hopkins shows that it really resets not only the tumor's metabolism, but the immune microenvironment. So the relationship between glutamine and FDG metabolism is actually quite important. So how do we do that? Well, here are some patient examples from a triple negative patient who had an extraordinarily hot FDG scan that had an SUV of 30. For those that are used to reading breast cancer, that's quite high. And for that amount of uptake, had relatively modest glutamine uptake. So fit the profile that we were expecting to see based upon our mice. And this has taken us to the point where we're ready to do something that was developed in our moonshot, and that is to image these two together. So what we can do is think about a 1 millicurie of fluoroglutamine followed by a 10 millicurie of FDG, relatively low exposure, and then use that to model a schema where we can do dual injections, starting with the glutamine, the reversible tracer, and then moving on to FDG, the trapped tracer. So again, this was work that Varsha Viswanath and Austin Pantel did together. They were able to optimize scan durations and looking for how to recover these parameters, and ended up coming with these relative doses that could be done in as short as a 20 to 25 minute scan, followed by a relatively short FDG scan. And in the space of 60 minutes, which was the space we used to do dynamics just for FDG, we could actually get both tracers at the same time. And so that's where I'm going to leave you, suspended in mid-animation. This little thing caused the pandemic interrupted us, and we're just about ready to take this into humans with this dual injection technique. So in my summary, why total body PET? Better pictures, better accuracy, whole body dynamic imaging. What's special? Shorter imaging, lower dose, whole body tracer kinetics. And when you can do this and you can use multiple tracers, you get more than twice the information by doing 2 or more tracers at the same time. So I'm going to stop here. I want to put up my acknowledgement slide again, and especially thank Joel and Austin and Rong and Varsha who contributed a number of slides here. Acknowledge my industry partners, my collaborators at University of Pennsylvania and elsewhere, and a number of funding agencies, including the Susan G. Komen Breast Cancer Foundation that helped support this. And so with that, I will stop, and I think I might have left a few minutes for questions. Thank you very much.